let's begin discussing the chemical senses. So from the name, chemical senses. So you know we're going to be detecting chemicals such as odor. Odor, right? That's a chemical. So the nectar of the flower, or maybe the perfume that you smell, those are chemicals. What about taste? Is taste not a chemical? So taste also, you taste something that's sour or something that's acidic or bitter or sweet. So those are chemicals. So today we're going to be learning how does your body process two chemical stimuli in the form of gustation. We said gustation is different from gestation with an E. So students sometimes tend to mix gestation with gustation. Gustation with a U, that is taste. And gestation with an E, that is pregnancy. Okay, so we're gonna see how your body processes. And you know, this diagram is going to be going with us all over the place. So we always going to have a receptor. We have a receptor. So if you're going to be discussing olfaction, smelling, or if we're going to be discussing taste, that one is going to be a chemoreceptor, right? Because these are chemical senses. So we can have a chemoreceptor. Then when we do the hearing, that's no more chemical. So we're not dealing with chemicals now. Now we're dealing with mechanoreceptors, sound waves striking and hitting the tympanic membrane and making some vibrations that I'm going to be able to translate. And then in the end, we're going to be doing the vision. And that is not going to be a chemoreceptor anymore. That's going to be a photoreceptor. But in the end, a receptor is a receptor. A receptor is going to be detecting. Sometimes I like to call the receptor a detector because it does detect a stimuli in one form of energy. You know, chemoreceptor, that's chemical energy. Photoreceptor, that's photo energy. You also have mechanoreceptor, that's mechanical energy. And we say the receptor is going to take the stimuli, it's going to detect the stimuli, then it's going to send an input signal. And we said that an input signal is always what? Sensory. Excellent. So an input signal is always sensory. Absolutely, completely sensory. There's never going to be an input signal that's motor. And the input signal that's sensory is carried by a special type of neurons known as the afferent. Excellent, excellent. Those are the afferent neurons. And then they finally lead us to the processing, integration, and control center. Processing, integration, and control center. And this processing, integration, and control center is going to process. And there's one more thing that happens here also. So when I'm about to make an output, I'm on the way to make an output. And we said that an output is always what? An output is always motor, excellent. And what's the type of the neuron that's going to facilitate for this motor output? So excellent. So that's going to be an efferent neuron. And the efferent neuron is going to take the output. There is something here. Remember when we were saying yesterday about how you take a peso or a Canadian dollar, but in the end, in Europe, they all have to be converted into euro. So here, my brain is gonna take a chemoreceptor stimulation, a photoreceptor stimulation, mechanoreceptor stimulation, osmoreceptor stimulation, nociceptor stimulation. But in the end, all of them are going to be generated in the same form. What is this process known as? So when you take different types of energies and then you change them, you convert them into a one standard currency that we all deal with in the body. In the body, we don't deal with different currencies. Like when you go to Europe, you have to have the euro, no matter where you're coming from to visit. So here, no matter where you're coming from, chemo, photo, mechano, you have to be converted into an action potential. What's the name of this process? Yeah. Transduction. Excellent. That's known as transduction. So the transduction, that's transduction. Students think that the receptor is the transducer. The receptor is the one that makes the transduction. It really isn't. It's your central nervous system that does this transduction. Your central nervous system, your brain, your spinal cord is going to be able to take these different types of stimuli and convert them into one uniform action potential. Electric energy. Even if it takes electric stimuli, it's still gonna respond as an electric stimuli. So that's your central nervous system being the brain and the spinal cord. By the way, we mentioned that the spinal cord in a couple of the diagrams, sometimes the spinal cord is working like a relay station, like as a relay station. You remember this diagram? So if you look at this diagram. Oh, amazing. Look at this diagram. In this diagram, the brain and the spinal cord, obviously, both are central nervous system. Those are processing, transducing, integrating, controlling. But what does the spinal cord do? It passes. Do you see that? Sometimes the spinal cord has its own way of responding, like the knee-jerk reflex. So when you hit the patellar tendon with a hammer, the spinal cord is not going to say, brain, what should I do? It's immediately the output is going to be the knee-jerk reflex. So that shows you here, the knee-jerk reflex is not going to have anything to do up here. The knee-jerk reflex is going to have an arrow pointing to the spinal cord and an arrow coming out the spinal cord. Does not involve the brain at all. But this one does involve the brain. So you see that the spinal cord sometimes acts as a processing center, but other times it passes the stimuli up to the brain. There is some type of neurons also that are up here. So they do not involve the spinal cord. They immediately take the information from the receptor, send it to the brain sends an output. Spinal cord has nothing to do with this. What's the name of these nerves? They don't even know what the spinal cord is. They have nothing to do with the spinal cord. They just go immediately, no passing, no they don't have to go to Grand Central and then take a subway somewhere else. They immediately go. What's the name of these nerves? First order. The first order. So the first order neurons are the ones that immediately take the stimulus and then pass it to the second order neuron that passes it to the brain, and then from the brain to the roof. You know, you have to remember the terminology. This cortex. Cortex means roof. You're gonna see it a lot. When we do the renal system, it's gonna be cortical nephron and medullary nephron. Medulla means in the bottom, in the core, down below. Cortex means the roof. So the most important part of your brain is the roof. That is where all you call the cortex. The cortex is the roof. So once you hit the thalamus is in the medulla. But once you go to the roof, that is your third order neuron. That's where you go to the final destination. It's the cranial nerves. These are the ones that have nothing to do with the spinal cord. No cranial nerve is going to take some photoreception or information from the eyes and say, let me go to the spinal cord, and then the spinal cord is going to pass me. No, they are already in the cranium vault. They are already up there in your head. So they don't need to go to the spinal cord. So now let us go and examine how this gustation happens. So gustation is a sensory perception of molecules that are dissolved in water. So that's the requirement. You don't taste as well when your mouth is dry compared to when your mouth is moistened with water. And there are different stimulation. When you have a meal, you can tell that was salty. You can tell if that food is bitter. You can tell if the food is sweet. And you can tell if the food is, there's a new sense called umami. Umami is like amino acid or protein, proteinaceous food like steak, any food that is rich in proteins, chicken or fish, or any food that has tissues, proteins, that's known as umami. We will see that. And there are about 4,000 taste buds, and they are distributed in different types of papilla. Some papilla do have a taste bud associated with them, and other papilla do not have taste buds. So let's look at the different types of the papilla. The papilla are these visible bumps that are found on the tongue. And by the way, taste is not only, is the food sour, is the food salty, is the food sweet, is the food umami? No, also the texture. Sometimes we enjoy the type of so taste is very complex. It's also the color of the food. That also has something to do with the taste. The smell of the food, that also is associated with the taste. One type of papilla is this lingual papilla, and these are found on the tongue, and these lingual meaning they are in the mouth. And there are 4 types. 3 of these 4 types of papilla are associated with taste buds, but one of them doesn't have a taste bud. These are the 4 different types of papilla. The filiform papilla, these are small spikes, and these are the ones that have no taste buds. So that makes it for an interesting question on homework, for example, or an exam. Which one of the 4 different types of lingual papilla doesn't have any taste buds associated with it? And you say, oh, that one is the filiform papilla. The foliate papilla, they form parallel ridges alongside of the posterior 2/3, and they do have taste buds. Fungiform papilla, they look like mushrooms, and each has about 3 taste buds. The vallate papilla, they look like a V shape. They are large papilla that are arranged in a V shape at the end of the tongue. These taste buds, they are clusters of 50 to 100 taste cells, and the taste cells, they are epithelial cells. They're not neurons, but they do communicate with the neurons. They send the information to the neurons. There are basal cells and supporting cells. So in every tissue, we have a parenchyma. So the parenchyma is the tissue that makes up our organ. And the stroma is the supportive tissue, that helps us accomplish the job. So the basal cells, these are the stem cells that replace the taste cells. So these are examples of regenerative cells. So taste cells, over time, due to the stress and due to the life cycle, they die and they must regenerate. And the cells which makes for an interesting question, is what's the name of the cells that are the progenitors that have the potential to develop and enter the cell cycle and replace those dead taste cells with new taste cells, and these are the basal cells. Supporting cells, they resemble the taste cells, but they have no synaptic vesicles or sensory role. So how do we taste? We taste by literally internalizing and taking in the food and bringing it inside the taste bud. And the taste bud is going to fuse those vesicles, those bubbles that are full of this food that's to be tasted, and then to send it to the brain, and the brain is going to be able to decipher, is this food salty, is this food sweet, is it bitter? And we said that the senses are important for our survival. So if you weren't able to taste, you cannot tell if the food is bad and poisonous and toxic. So taste helps us to tell if the food is fresh so that we can continue, or if the food is nasty, we should abandon it. Okay, so there are 5 primary sensations. Salty sensation, and that is the electrolyte. So, you know, electrolytes are important. We discussed that we want a fixed homeostatic range of electrolytes. We have a certain amount of calcium, certain amount of sodium, certain amount of potassium. We don't want hypercalcemia. We don't want hypocalcemia. We don't want hyperphosphatemia. We don't want hypophosphatemia. We want U. We want U range or homeostatic range of the electrolytes. How do we tell how our electrolytes arrange in our body? That is with the salt. So during pregnancy, mothers are always deficient in minerals. So they're always craving salty food. And sweet food is associated with carbohydrates. So carbohydrates and foods that have high calorie count, they always taste sweet. And we said this word here, umami, which translates to delicious in Japanese, because that is where these tastes were discovered. That is for all the proteinaceous food, like meaty or savory taste of the amino acids in the chicken or the beef broth. Sour food, that is acidic, and bitter is basic. So you have to know that one. You have to know that your body is able to tell if the food is sour because it is acidic. And if the food is bitter, that means it's basic. And we have a very low threshold for bitter food compared to sour. What does that mean? So that means we are excellent at detecting bitter food way more than our ability to detect sour food. Why is that? Because bitter food is poisonous. So you need a very little threshold of bitterness, and then you will immediately know something is wrong. And that is actually good for the homeostasis, for the well-being, and for your health, because you cannot tolerate. Imagine the bitterness threshold was really high, so you would be intoxicated. You'd be poisoned long before you detect the bitterness. Is that clear? Questions? Okay. So more taste is being discovered, actually. So the taste buds are being studied, and we are constantly discovering different types of oligustus. Oligustus, these are different receptors. These are different taste buds that are able to help us, tell what kind of food that we're eating. And taste is influenced by, as we said, the texture, the color, the appearance, the temperature. So these things influence your experience when you are having a meal. Sometimes some food, you really cannot eat it unless it's hot. And sometimes when you look at the color of the food, it looks really appetizing. And so now when we look at the physiology of the taste, there are two mechanisms that help us taste. The first one is if the food that we're eating is hydrophobic. If the food that we're eating is hydrophobic and the food is hydrophobic, it cannot be, internalized. So your body is able to, your taste cells are going to be taken fragments. So that point we discussed. How do your taste cells work? They take fragments of the food that you internalize in your mouth and they bring it inside into your taste buds. And we see there are two mechanisms. So sugars and basic food and glutamate, they activate G protein coupled receptors. So I think we should have a background on what the G coupled protein receptors do. These G coupled protein receptors are dealing with a hydrophilic substance. So hydrophilic substance is a substance that can cross the plasma membrane or cannot cross the plasma membrane. Hydrophilic. Can hydrophilic substance cross the plasma membrane? It cannot, because the plasma membrane has a lot of phospholipids. So it only hydrophobic substance, like hormones that have cholesterol in them, like testosterone and estrogen, progesterone, they can easily cross through the cell membrane. But hydrophilic substance, like these catecholamines, like epinephrine, as we will see, these hormones are unable to cross the plasma membrane, which is nonpolar and hydrophobic. So we utilize this system. So you're gonna see this system a lot. This G protein coupled receptor. The G protein coupled receptor operates by binding on the outside. So you have your, let's assume that this is the plasma membrane. The plasma membrane has polar heads, and it has non-polar tails. Polar heads and non-polar tails. If you are non-polar, you can easily cross through the plasma membrane. If you are non-polar, like steroid, if you are a steroid, you are welcome to cross through the plasma membrane. This hydrophobic butter, this is butter layer, non-polar, phospholipid, hydrophobic tails, hydrophilic heads. But if you're hydrophilic, like the food that you're eating, some food that you eat is hydrophobic, can be internalized. But some food that you eat is hydrophilic, like glutamate, carbohydrates, and alkaloids. So how do they get it internalized? How do we taste them? So there are receptors on the cell surface that are bound on the inside to a G protein. So they are coupled from the inside to a G protein. That's why we call them G coupled protein receptors. So when this hydrophilic food is binding to the receptor on the outside, that's going to cause a conformational change on the receptor. The receptor is going to dimerize, and that is going to make the G protein active. When the G protein is activated, the G protein is going to go and is going to initiate a cascade. Many enzymes that were off are not going to be turned on. Many enzymes that were off are not going to be turned on. And in the end, we don't want to go into details, but in the end, we're going to have a target effect. We have a target effect after we utilize a second messenger. So the first messenger is the signal. The first messenger is the gustatory stimulus. What we just took in our mouth, the sour food, the salty food. So if it's a carbohydrate, if it's an alkaloid, if it's glutamate, it is hydrophilic. It cannot cross. So it will end up binding on the outside and then a second messenger. That's the most important point. A second messenger is going to carry the signal and is going to lead to the target effect, which is taste. We're going to be seeing this a lot. We're going to be seeing the G protein coupled receptors operate in different modalities. We see it now in taste, but we're going to see it in the future. So is this point clear? Is this point clear that when we have any substance that is hydrophobic, it can cross right through the taste bud. If you're eating something hydrophobic, the taste bud can internalize it easily. But if you're eating something hydrophilic, catecholamine, such as the sugars, the alkaloids, and glutamate, we must utilize the G-coupled protein receptor system, and a second messenger is going to lead to a target effect. Is that clear? Okay. Okay, projection pathways. So remember, we always, when we're doing sense, we're going to begin number one with a receptor. That's the first step. Receptor detecting a stimulus. Number two, the input, the sensory, the afferent neurons. These are the projection. These are the representations of the projection pathway. So this projection pathway is now, so when we eat the food, what are we trying to do? We're trying to figure out, is it salty? Is it sweet? Is it bitter, meaning it's poison and toxic? And we need to, not eat it because it's gonna cause us sickness and disease. So projection pathways are always leading to the central nervous system, leading to the brain. So now let's see how is food being tasted. So there are three cranial nerves that carry the taste information. We have the seventh cranial nerve, the facial nerve. It's responsible for detecting stimuli from the anterior two-thirds of your tongue, while the glossopharyngeal nerve is responsible for the posterior one-third of the tongue. And then if there's any food remnants on the palate, so you don't only taste with the tongue. Sometimes there is a taste sensation in the palate, in the pharynx, and the epiglottis, and who relays that information is the vagus nerve. And so, and remember, those are cranial nerves. So we're not involving the spinal cord. We're directly going to the brain. So you see, the facial nerve is going to be taken two-thirds of the stimulation from the tongue to the brain. Glossopharyngeal nerve, that's the afferent nerve. You see, those are afferent nerve. Those are projection pathways. Glossopharyngeal, it will take posterior one-third and send it to the brain. And if there's any food scattered in the roof of the palate and the pharynx and the epiglottis, the vagus nerve is there, and it's going to be taking everything and sending it to the brain. So fibers from all these nerves are going to synapse in the solitary nucleus of the medulla. So you see now where we're going to end. So that's just for taste. So we're going to go to the solitary nucleus of the medulla. So when we get to the medulla, that is where we integrate. Then the second order neurons. So that's the first order neuron, the facial nerve, the glossopharyngeal nerve, and the vagus nerve is heading as your first order neuron. The second order neuron is going to go from the medulla, depending on what type of food are we dealing with. The second order neuron is going to communicate between the medulla and the hypothalamus and the amygdala, or the thalamus. And then from the thalamus, we're going to go to the primary gustatory cortex. So at the roof of our brain, we have the gustatory cortex, the computer cortex that is responsible for food detection. And lastly, there's the third pathway, which is the thalamus, that's going to be relaying the signal to the orbitofrontal cortex. And these are different terminations based on what are we trying to taste. So you're gonna go from the solitary nucleus of the medulla into one of those three pathways. Okay, so that was taste. So now we're gonna look at olfaction, which is smell. So if we detected tastants in order to detect taste, so tastants are chemical substance that elicit taste. We have odorants. So those odorants are responsible for eliciting smell. So we have olfactory mucosa. Olfactory mucosa is a paste or a patch of the epithelium that is found in the roof of the nasal cavity, and it houses those receptor cells and receptor hairs. So you see here, this is the cribriform plate, and you see that that is your respiratory mucosa. And the same way we internalize food substance inside the taste bud, we also internalize those odorants and bring them into vesicles that are later on going to be deciphered into the brain. So that's the pathway here. So if the air is flowing inside your nose, there is a paste of mucosa, and this mucosa is made up of olfactory hairs. These hairs are going to be detecting and trapping. They are like glue. Mucosa is like glue. They're going to be trapping and detecting this odor, and then it's going to be passing it to olfactory hairs. From olfactory hairs, we're going to be going to olfactory bulbs, olfactory cells, and then these basal cells are the stem cells that are going to differentiate to replace any damaged olfactory cells. Then we're going to enter through the cribriform plate. This is a bony structure, and from there we will have an olfactory nerve, and the olfactory nerve is going to be carrying this odor that is going to be relayed, finally terminating in the olfactory bulb. And from the olfactory bulb, there is a cortex in the roof of your brain that is responsible for detecting the type of odor that you're taking. And we said that odor is very important for both, for survival and for reproduction. Odor is responsible for detecting the good odors that are going to guide you to eat something that's healthy, or to detect the smell of something that's dangerous so you can remove yourself from that region and survive. Also for reproduction, those hormones, females during the menstrual cycle, during ovulation, their sense of smell tends to become even more sensitive than before. That was found in research studies. So you see there is variation. There is gender variation in senses. There is individual variation. So in the lab, one of the questions, remember, we asked yesterday is, did we get different responses in the way that we tasted had we done the actual taste? But you would have seen that people would taste something more sour than others, and there is a spectrum in your responses to this taste stimuli. Questions? So the same way when we dealt with taste, we utilize the G protein coupled receptors in taste. We also utilize G protein coupled receptors in odor also. So whenever you see a G protein coupled receptor, what does that indicate? It indicates we're dealing with what kind of chemical substance? So G coupled protein receptors, those are G coupled protein receptors. Those are activated when we are dealing with a chemical messenger that is unable to do what? Why do we have this system of G coupled protein receptors? Exactly. So whenever you see G protein coupled receptor, you rest sure that you're dealing with some chemical substance that cannot cross, that cannot enter. So somebody, a second messenger on behalf of the first messenger is going to lead to the target effect. So sometimes you're smelling an odor that is hydrophobic, it can enter through the cells. But if you're smelling or sensing something that is hydrophilic, then a second messenger is going to relay the signal. What's the first messenger? What's the first messenger? The first messenger is the one that's unable to enter. So that is your tasting, the thing you're trying to taste, but it's hydrophilic. That is your odorant, what you're trying to smell, that is also hydrophilic. So the first messenger is what you're detecting, is what the receptor is detecting, but it cannot internalize it and bring it inside due to its chemical structure being hydrophilic. So whenever you see the G protein coupled receptor, you know you're dealing with a hydrophilic substance. Who is the second messenger who is going to do the job? So you know, if let's say the school is closed, you know, you cannot enter the school unless you have an ID, and you call for Uber. That Uber driver cannot enter to inside the school. They must contact public safety, for example, and the public safety is going to be the second messenger who's going to come and get you or deliver the message to you. But if the Uber driver, him or herself were LIU student, they can themselves walk into the building. They don't need a second messenger because they are also part of the structure. That's exactly what happens when you're dealing with steroid hormones, steroid substance, hydrophobic substance. They can easily cross through the plasma membrane. They don't need a second messenger. Testosterone does not need a second messenger. Estrogen does not need a second messenger because they are made up of lipids. They're made up of butter, just like the cell plasma membrane. Who needs a second messenger? These are the polar hydrophilic substances that are, for example, epinephrine, the fight or flight hormone. And also, since we're doing general senses, the salt is hydrophilic. So that one needs a second messenger because it cannot cross the plasma membrane. But if you're eating something umami, such as you're eating steak, that one does not need a second messenger because it can be internalized immediately through the plasma membrane. Is that point clear? Okay. Second messenger is always known as CAMP, cyclic AMP, cyclic adenine monophosphate. So if you're dealing with a G protein and the G protein is gonna facilitate for a second messenger, the second messenger is always absolutely cyclic AMP. So we take ATP, adenine triphosphate, and we devoid 2 phosphates from it, we end up with mono, 1 phosphate. Then we cyclicize that, we end up with our notorious cyclic AMP, and that is going to be our second messenger. Now we're going to look at so this was the process of detection. So since we are firstly detecting the odor by detecting odorants, or taste, we are detecting tastings. With odorants and tastings, we detect them based on, are they hydrophobic? Then they will be utilized as first messengers. First messengers that will elicit the odor response and the taste response. But if they're hydrophilic, then that means we're gonna need a second messenger. And what's the name of the second messenger? What's the name of the second messenger who will be activated whenever we're dealing with a chemical substance that could be an odorant or a tastant that cannot be internalized because it's hydrophilic? Who is the second messenger who's gonna be operating with the G coupled protein receptor to elicit the response? Yeah, so cyclic, so we're gonna be using cyclic AMP to relay the signal, and then we're done with reception. So this process here is simulating the process of reception. So now we're gonna look at the projection pathway. The same way we looked at the odor projection pathway, now we're gonna be looking at the taste projection. Now we're gonna be looking at the odor projection pathways. Okay, so let's look at the olfactory or the odor projection pathways. So the olfactory cell axons are going to be passing through the roof of the nose, through the cribriform plate, and they're going to enter pair of the olfactory bulbs, and then they will synapse with dendrites of what's known as the mitral cells. Then there are tufted and mitral cell axons that will carry the output from the glomeruli, and then they're gonna send through olfactory tracts. These olfactory tracts are going to be delivering the odor that has been detected. These are the olfactory tracts. These are the projection tracts or the projection pathways that are going to be delivering the odor to be processed and transduced in the brain. Is it helping for our survival? Is it helping us in reproduction? All of that is going to be carried through the olfactory projection pathway. So if we examine this process here, how does the olfactory projection pathway operate in the brain? So the first step, the olfactory signal that has been detected by the olfactory receptors in the cribriform plate. The olfactory signals are going to be received in these olfactory bulbs, and then the signals are going to be relayed caudally in the olfactory tracts. So that's the second step. The olfactory projection tracts are going to be carrying the olfactory signals, and then the primary olfactory cortex that is found in the temporal lobe. Remember, we have a frontal lobe, and we have the occipital lobe, and we have a temporal lobe. So your olfactory center for deciphering olfaction is found in the temporal lobe, and that is going to create a conscious perception of the odor, and that's going to relay the signals to other parts of the brain. Signals from each temporal lobe are relayed to the contralateral temporal lobe. So all the processing is mirrored in both cerebral hemispheres. And both cerebral hemispheres are going to cooperate in order to help us, integrate and process what is the nature of this odor that we have detected. So signals relate to the amygdala, and that is the center for survival, to detect if this odor is going to reflect that this is a dangerous situation. The hippocampus, the insula, and the hypothalamus, they're gonna relay this odor to the olfactory memory, and they're gonna produce an emotional and visceral response. And lastly, signals that are received in the cortex are going to be combined with other information to identify and tell the difference with relation to what kind of odor have we detected. Then feedback to the olfactory bulb is going to modulate the perception of the odor according to circumstances. So what does that mean? That means the last step when we feedback. So first of all, we take an odor, and then we internalize it, and then we decipher, is it a healthy odor? Is it promoting survival? Is it promoting reproduction? Then we associate it with an emotion and a memory. And then after that, we feed back into the olfactory bulb. Sometimes the reason why you no longer can smell something is because step number 7 took place. So there's a feedback from the olfactory cortex telling your olfactory bulb, your olfactory receptors, that we already know that this odor that you have sent us through the projection pathway is safe. So it's not something that is causing an alarm. So that's why you adapt. It's not going to trigger. It's not going to trigger any, any more internalization. It's not going to trigger any more sensation, because we already figured out there was a success. We detected the stimuli, we sent projection pathway, and the projection pathway in your olfactory cortex realized and attached that odor to an emotion, and attached the odor to an emotion and a memory. And then a response was sent back to the olfactory bulb, telling the olfactory bulb that you are in a safe place, and this is a good, probably a good smell. Or if it's something that is reflecting as danger, it's going to feed back and cause you to remove yourself from that particular place. So the most important step is 7. 7 is where we find out. Am I smelling something that is dangerous, that is threatening my survival? Or am I smelling something that is promoting my health and my well-being? And then, when you no longer can smell it, it's because your brain has already approved that this is something that is not threatening your survival. Questions? Questions? Okay. Okay, hearing and equilibrium. Something that is very interesting that we discussed in the past, and that is your ears in the first place, their job wasn't actually to listen or hear anything. What was their initial job? Your ears. Yeah. So your ears were responsible to make sure that you are standing upright and when you walk, you are balanced. And that's why babies, they have a trouble in the beginning walking, because the development of their inner ear is actually still, developing. So in their developmental biology, it's going to take them a while to develop their organ systems. And once the inner ear has fully developed, that is when the baby is finally able to walk balanced. So it's not because the muscles, the baby's muscles are too weak to hold the body, or the baby is not strong enough. It is because the inner ear hasn't actually developed completely. Questions? Okay, so hearing is the response to vibrating air molecules. Is this chemical? Is this anything chemical? Vibrating air molecule, that is not chemical. What is that? Is that electrical? That is mechanical. So that's a mechanical sensation. Oh, it's hot. And today is very bad, actually. It's really hot today. Okay. Equilibrium is the sense of motion, body orientation, and balance. So both senses reside in the inner ear, which is a maze of fluid-filled passages and sensory cells. So the fluid is set in motion, and the sensory cells convert this motion into an informative pattern of action potentials. So this sound is any audible vibration of molecules. Sound can travel in solids. Sound can travel in liquids. Sounds can travel in air. Sound can never travel in what? Extra credit, if you can figure that one out. Space, very close. Space, vacuum, that's the word I was looking for. Okay, you guys are gonna split the extra credit. So half. Excellent. No sound can transfer in vacuum, guys. No sound can transfer in space. No astronaut took off the helmet and said to his astronaut, "Good morning," and that never happened, actually, because sound does not pass in vacuum or in space. Sound only needs a medium to carry these waves. So the waves need somebody to carry them, guys. Vacuum is empty, so it's not, so you're not gonna hear anything in vacuum. So the physical properties of the sound. We discussed how salty the food is. There is a understanding of the intensity of the stimuli. Some foods are more salty than others. Some foods are more sour than others. Some sounds are louder than others. Sometimes the pitch is high.
which represent the frequency of the vibration. And loudness is the perception of the sound energy or the intensity or the amplitude. Normal conversion has a loudness, normal conversation has a loudness of 60 decibels. The threshold for pain is 120 to 140 decibels. Ever had headphones and you've been listening for a while, then you had ear pain, especially with those really big headphones? You know, the anybody had that experience? Never. You never had headphones on and then your ears hurt. Yeah, I had that a while ago. So, they say if the sound is too loud and that means you're too weak. Actually not. It means you're gonna rupture your tympanic membrane. So all of this really loud stimulation is gonna become stressful because we don't want hyper vibration. So this hyper vibration is going to cause some form of stress that's gonna damage irreversibly, going to irreversibly damage the tympanic membrane. So this figure here shows us the range of the human hearing. So we see that how do we interpret the sound based on the frequency and the loudness? And if we want to understand how the ear operates into the reception of sound, so this is always going to keep repeating this diagram here. Reception of sound, projection of input sensory afferent sound waves. Then we're going to be processing, integrating, controlling, transducing, figuring out, the sound, a language. You hear the language, your brain is going to be deciphering. What is that? You hear the sound of a lion, you hear that's something that's really dangerous. You want to survive. You hear the sound of beautiful birds, that's gonna make you happy. You're gonna walk towards that sound. And then an output is going to be generated. So now, in order to understand how the ear works, we have to look at the structure of the ear. Remember, we said the knife is sharp, that's why it's able to cut. Had we not known that the knife is made up of a sharp metal, we would have never found out that it's able to cut. So let's see how the ear is formed. This outer external ear looks like a funnel, actually. This auricle is made up of cartilage. The upper part of your ear is made up of cartilage. What about this bottom lobe? What is this lobe made up of? That's not cartilage. The one where you put the earring. That is not cartilage. What is that? What could that be? Connective, yeah, it's connective tissue. But what kind of connective tissue? Because cartilage is also connective tissue. When you say extra credit, the attempts come out. Okay, another extra credit. What do you think that is made up of? That is not cartilage. I already told you. So it's a free guess. What do you think that's made up of? Touch it. Touch it, and it may give you a clue. Fat. That is fat. That is not cartilage. So it's pretty interesting how the ear is made up of the whole outer part is cartilage, and the lobe, the ear lobe, is not cartilage. That is fat. And you can tell by sensing, by feeling. Yeah. Okay. All righty. Cerumen wax. What is that? So this ear wax, originally, it is made as a immune system substance that is supposed to trap, like the mouse trap is going to trap the mouse. It's supposed to trap all of the debris in the air. And once it binds with the air, it's going to turn into wax. So on its own, it's really not a problem. It's not going to impair your hearing. But if you have a lot of earwax, it means maybe your job is you are spending a lot of time in places that are full of debris or pollutant, and these pollutants are entering into your ear, and that is what causing the cerumen to clog your ears. But it's responsible for trapping microbes, and it keeps the eardrum pliable. Okay, so that's the Earlobe, we said that part is fat, but everything else is cartilage. Now we go from, so that's the outer ear, you can see the outer ear. And we said that the outer ear is important because what does it do? It looks like a funnel, doesn't it? So it funnels the sound, it focuses the sound, okay? And the middle ear is made up of, is located in the tympanic cavity in the temporal bone, and it is bounded by the eardrum and the inner ear. There is a tympanic membrane and the tympanic cavity. The tympanic membrane is known as the eardrum. So sometimes we refer to anatomical landmarks with colloquial terms. Remember, the kneecap. What was the kneecap? That's your patella. That was your patella bone. That's the kneecap. What is the collarbone? Collarbone. What is that? That's the clavicle. And what is the skull cap? That is the calvarium. What else? The scapula is known as the shoulder blade. The tympanic membrane is known as the eardrum. So those are very important anatomical landmarks that have two names that help us understand their structure. So this tympanic membrane is going to be the site where we receive the sound vibrations. And it is not just a membrane, it is pasted with nerves. Why? Because the nerves are gonna take the mechanical vibrations. Because remember, we don't only have chemoreceptors and photoreceptors, we have mechanoreceptors. So that's why they are stuck to those nerves are stuck to the tympanic membrane. So when the tympanic membrane is shivering, when the tympanic membrane is bouncing, those nerves are able to be stimulated. And then they get to send a projection pathway. Are we clear? Okay. Then we have these auditory ossicles. Those are the smallest bones in your body. These are the three smallest bones of the body, and they connect the tympanic membrane to the inner ear. So the vibration that takes place in the eardrum is going to be propagated and carried to the inner ear through these three bony, smallest bones in your body. The malleus has a long handle attached to the inner surface of the tympanic membrane and a head that articulates with the incus. The incus is going to receive the stimulation. It's like the dominoes effect. So the malleus is going to tip over and stimulate the incus. The incus has a triangular body that articulates with the malleus, and the long limb is going to articulate with the stapes. And the stapes is going to receive and propagate that mechanical vibration. So it's shaped like a stirrup. It has a base known as the foot plate that is held in an opening called the oval window. The stapedius muscle is attached to the stapes, and the tensor tympani muscle is attached to the malleus. So here we see the malleus, the incus, and the stapes. Once the tympanic membrane is vibrating, we have the domino effect. We tip over the malleus, then the incus is gonna tip over and it's gonna touch the stapes. Then the stapes are gonna be transferring this vibration, this mechanical stimulation to your inner ear. So we are very concerned with middle ear infections known as otitis media. And we know whenever you end with itis, whenever you end up with itis, you know that's a form of an inflammation. And an inflammation is caused by different triggers. One of them is an infection. What's the difference between an infection and an inflammation? What's the difference between an infection and an inflammation? So all roads lead to Rome. Inflammation. So there are different But you know, inflammation is not triggered by something good. So very negative things cause inflammation. Very negative, bad things cause inflammation. Trauma, like a contusion or a bruise. A contusion or a bruise, is that a good thing? That's a negative thing. That's going to lead to inflammation. What else leads to inflammation? Trauma or physical agents like hyper or hypothermia. If you have hyperthermia is too hot or hypothermia is too cold, that's going to lead to inflammation. And also you have infection. What's an infection? Infection is when microbes are bacteria, fungi, virus, parasite. When they attack you and enter your body, that's an infection. No infection without a microbe. Nobody said it was so hot outside, I had an infection. No, you get an infection when there is a microbe. But all of them lead to inflammation. Inflammation is when there is 5 things: redness, heat, pain, swelling, and loss of function. So whenever you have an inflammation in one place in your body, you're going to be feeling redness. If anything in your body is red, anything in your body is hot, anything in your body feels pain, anything in your body is swelling, if anything in your body lost its function, then it's undergoing inflammation. Inflammation, even though it's red, pain, hot, swelling, loss of function, is actually a good thing because that's how your immune system is able to fight the infection. That's how your immune system is able to restore your health. And then after the combat, so inflammation is a combat when your body is fighting the microbes, and then after the microbes have been removed, and also the dead cells, the dead or damaged cells have been removed, then we begin healing. We begin healing. If the tissue is regenerative, not all tissues enter the cell cycle. You're supposed to take that in A&P1. Some tissues, they enter the cell cycle and they can regenerate. So yeah, like your skin. You cut your skin, new skin is gonna come out. You cut your brain, no more brain is coming out. That's why you're never gonna have a stroke in your skin, or you're never gonna have a heart attack in your skin. Because what's a heart attack? What's a stroke? Stroke is when nerve tissue died. Heart attack is when cardiac heart tissue died. But skin cells don't die, because once they flake off, they exfoliate and they die, new cells are gonna regenerate. So some tissues can regenerate, can birth again. And some tissues, once they damage, they die, they can never enter the cell cycle again. So after inflammation, if the tissue is regenerative, we heal by regeneration. We enter the cell cycle and we make new cells. But if the tissue is non-regenerative, what happens? If the tissue cannot, like for example, like we said, the skin, you can have more skin. Your bone, you break your bone, new bone is gonna form. But we said some other type of tissue that once it's dead, it cannot heal, like your heart, like your brain. So what happens then? Fiber. You have fiber deposition, fibrosis. You have fiber. We're gonna talk about that. Fibrosis, fiber formation. So otitis media is common in children because the auditory tube is so short. And you don't want an infection in your middle ear because the infection is gonna spread into your brain. Your infection is gonna spread into the meninges, and that's very dangerous. So the infection is easily going to spread from the throat to the tympanic cavity and the mastoid air cells. Symptoms of the middle ear infection are when the fluid accumulates in the tympanic cavity, which produces pressure, pain, and impaired hearing. And we sometimes perform a surgical operation known as tympanostomy. What is tympanostomy? Tympanostomy is the lancing tympanic membrane and draining the fluid from that tympanic cavity. So you see, that is the auditory tube for a child and the auditory tube for an adult. What do you think is the difference? What do you see here is the difference between, so you're just looking at the auditory tube of a child, pediatric, and the auditory tube of an adult. What do you see? You see that the auditory tube is shorter. That's why it's very dangerous for children. It's common in infant because their auditory tube is short and horizontal. Excellent. So it's very easy to spill over all of the infection material into your brain. Then you have your inner ear. So a portion of the ear is housed within a maze of temporal bone passages, and you have the bony labyrinth, there's a membranous labyrinth, and there's the vestibule. The vestibule is the chamber where the labyrinth are going to begin. So we have the endolymphic sac and the posterior lateral and the semicircular. See these semicircular ducts, these fluid-filled semicircular ducts, semicircular canals, and they are surrounded by the temporal bone. So to house them from damage, see these circular fluid-filled canals, they are susceptible to trauma and damage. That's why they are surrounded by this bony structure known as the temporal bone. And you know the bone is always protecting viscera. So your lung and heart are protected by your rib cage. Your bladder is protected by your pelvis. Your brain is protected by the cranium, by the skull. So always the bones are important to protect very fragile, vulnerable structures. So you see these blue circular canals, they are all surrounded by the bone, the temporal bone. But severe trauma to the temporal bone, this boxing sport or getting hit in the head or blunt trauma is still going to overwhelm the capacity of the bone to fend off any trauma, and that's going to cause a defect into the circular canals. The inner ear is made up of the cochlea, and that is the organ, that is the main essential, that is the essential organ of hearing. And you see that these circular canals are made up of the scala vestibuli filled with a fluid known as the perilymph, and there is the endolymph that is found within the cochlear duct. The inner or the internal ear is a spiral organ that has an epithelium composed of hair cells and supporting cells. There are two types of hair cells. There are inner hair cells and outer hair cells. Inner hair cells are single rows of about 3,500 cells, and they are responsible for the sensation of hearing. But the outer hair cells, there are about 20,000 cells, and they are responsible for not the actual sensation of hearing, but they are responsible for adjusting the response of the cochlea to different frequencies and to increase the precision. So the outer hair cells are important for fine-tuning the sound that has been detected and sensed by the inner hair cells. So that shows you the inner hair cells, and that is the outer hair cells. So how does the physiology of hearing take place? So there's a contribution that comes from the middle ear. So the tympanic membrane is going to move in the air and is going to vibrate easily. Once we detect the sound, there's going to be a vibration in the tympanic membrane. It has 18 times the area of the oval window. The ossicles are going to concentrate the energy of the vibrating tympanic membrane on an area of 1/18 of that size. The ossicles are going to create a greater force per unit area on the oval window. And the ossicles and their muscles have a protective function, which is to lessen the transfer of the energy to the inner ear. The tympanic reflex is very important because loud noise is going to cause the tensor tympani to contract. And that's going, this contraction of the tensor tympani muscle is going to tense the eardrum, and the stapedius muscle is going to contract, which is going to reduce the vibration of the stapes in the oval window. Then the cochlear hair cells, these are the main essential organs for hearing. Their vibration is going to be passed, like we said, as a domino effect to the ossicles, the smallest bones in the body. And they're going to cause pressure waves in the inner ear fluid, and movement of the inner ear fluid is going to cause the vibration of the basilar membrane, and the hair cells are going to move with the basilar membrane. The structure and arrangement of the stereocilia of the inner hair cells, and they are bathed in high potassium fluid known as the endolymph, creating the electrochemical gradients from the endolymph to the hair cell cytoplasm. The single transmembrane protein at the tips is going to function as a mechanical gated ion channel, and the stretchy protein filament is going to connect the ion channel of one stereocilium to the sidewall of the next. So this is also similar to what's happening in the smallest bones of your body, these ossicles. So this domino effect is going to transfer now from bony structures into these hair structures full of potassium. So the tallest stereocilium is going to be bent. So we are seeing this bent happening, this bending happening from the tympanic membrane. So the tympanic membrane is going to be stimulated to pass this vibration, and then the vibration is going to be passed over to the smallest bones, and the smallest bones are going to carry this vibration to the stereocilia that are viscera full of potassium. So that is what we're talking about. You see the domino effect. Okay, is this concept clear? We have a passing, a process of progressive passing of vibrations that begins in the outer ear. And you have the sound waves, and the sound waves are going to strike and hit the tympanic membrane, and the tympanic membrane is going to progressively pass this vibration movement through these three bony ossicles, the incus and the stapes. And then finally, we're going to transfer into the stereocilia that is fluid filled with what electrolyte? What is the electrolyte that is filling this fluid? Potassium. The tallest stereocilium is bent when the basilar membrane rises up toward the pictorial membrane. Pulls on the tip lengths and opens the ion channels, and potassium is going to flow in. When potassium flows in, which is quite different from your traditional action potential, there's a depolarization that causes the release of the neurotransmitter, and that's gonna stimulate the sensory dendrites and generate an action potential in the cochlear nerve. The cochlear nerve, once an action potential has been generated in the cochlear nerve, then you're gonna be able to hear, because the cochlear nerve is what's going to be stimulating the hearing process. So nerve deafness. So somebody who's suffering from nerve deafness, it's because the cochlear nerve has been damaged. So no relay, no more relay of action potentials through this cochlear nerve. It's very hot. Do you see the domino effect happening again? But this is not happening in the outer ear. It's not happening in the middle ear. Where is this happening? That's happening in the inner ear. So they are bent, and once they are bent, which electrolyte is influxing? Which electrolyte is entering into the core of the cell? What's the name of the electrolyte? Potassium, and potassium leads to? Remember we said the traditional action potential begins with an influx of sodium, and that causes the depolarization. Except in hearing, there's no sodium. Who's causing the depolarization? The influx of potassium. Excellent. Very good. Once we have now detected this sensory stimulation, now let's look at the coding. Variations in the loudness or the amplitude are going to cause variations in the intensity of the cochlear vibrations. And the pitch is going to depend on which part of the basilar membrane is going to vibrate. So these vibrations are constantly being converted into depolarizations. Mechanical vibrations are converted into potassium influx signals and depolarizations. Then we said the outer hearing, the outer hairs, the outer hair cells are responsible for the fine-tuning of this sound that we have detected. And once the sound has been detected, now we move on from the stage of perception, from the stage of sensory detection, to the stage of auditory production and auditory projection. So now we are carrying an input sensory hearing signal to where? If we have an auditory projection pathway, we have finished with this step of coding a vibration stimulus with the domino effect that's gonna terminate in the influx of potassium and depolarization. So now we have generated a depolarization that was based on a sound wave. That depolarization is not based on a taste of some chemical. That depolarization is not based on the smell of some chemical. That depolarization is based on the influx of potassium due to vibration. So we're done with that. Now we're doing projection. So we're gonna project to who? Who are we projecting to? Since we're dealing with hearing, we're projecting to which roof, which cortex? Yeah, so the cerebral cortex is a big word. It's like the airport. But which terminal are we going to? Auditory. We're hearing, so that's an example of auditory cortex. So we're gonna be heading to the auditory cortex. So you're gonna enter the airport, but you're gonna go to the auditory terminal. And what's the auditory terminal? The auditory cortex. When we do vision, what are we gonna project to? Which cortex are we going to project to? Visual cortex, visual cortex. So the cortex is the big, really big airport. Which terminal are you headed to is what your signal is trying to get to. Okay, the second order neurons. These are cell bodies in the cochlear nucleus, and some fibers ascend to the. So the first order neurons, you know, the first order neurons are immediately from your inner ear, and they terminate. So the axons that are going to lead away from the cochlea, from your inner ear, as the cochlear nerve. And they're going to join with the vestibular and the vestibulocochlear nerve, cranial nerve number 8. And each ear is going to send nerve fibers to both sides of the medulla, and they're going to synapse with second order neurons. The second order neurons, these are cell bodies in the cochlear nucleus. So some fibers are going to ascend to the superior olivary nucleus, So that's the destination of your second order neuron, the superior olivary nucleus. And the third order neurons are gonna go from the superior olivary nucleus into the thalamus. And then from the thalamus, we're gonna head into the auditory cortex. So the auditory system has extensive decussations, so damage to one side of the cortex does not cause unilateral hearing loss. So this is our very interesting diagram. We see here that we have the auditory pathways in your brain. It looks confusing, but not really, because you know this is the termination site. Primary auditory cortex, that's the terminal of your hearing. And auditory reflex head turning, that is the outer response, that's the final response. And those two, the green center and the blue center, the green center is the inferior colliculus of the midbrain. And the blue center is your superior olivary nucleus of the pons. So they receive, so the arrow has to actually head towards these centers. So the story begins here from the cochlea. The cochlea is going to send through the vestibulocochlear nerve, cranial nerve number 8, its electrical action potentials to the cochlear nuclei of the pons. That's the first order neuron. Then from the pons, we're going to head into these two nuclei. And lastly, we're going to terminate in the primary auditory cortex. We're also going to have a mechanical response known as the auditory reflex, the head turning. You know, yesterday we discussed when we were doing in the lab that supposedly you're supposed to smell ammonia and your head is supposed to bounce back. Why is that the case? You know they do this to somebody who fainted. If you fainted, you lost consciousness, you smell ammonia, you wake up. So that's a response to an abrasive or something that is your brain is responding to something that is threatening to your survival. So ammonia is not pleasant. So the reflex of the head turn, remember the one you were looking for yesterday, that was to show you that there are unconscious reflexes that are promoting your survival. If you smell something that is beautiful, it has an emotion of happiness. If you smell something like the ammonia, it's gonna trigger shock. And it's gonna be so shocking that even if you were fainting, it's gonna make you wake up. Ever encountered that before? That somebody who fainted, usually people make them smell ammonia that can restore consciousness. Yeah, if they are not suffering from coma, because if a patient is suffering from severe coma, no ammonia can wake them up because now there's a damage to their balance and consciousness center. Okay, that's the first order neuron. Second order neuron in the pons, then the thalamus, third order neuron, and then finally the fourth order neuron. See the roof? The outside is always the roof. Outside is always the roof. That's the most important part of your brain. All of the centers are at the roof, and all of the final neurons are terminating in these roofs. Which roof are we dealing with? The auditory cortex, the visual cortex, and all of the centers that are responsible for your survival are found in the cortex. Because cortex means outside roof, while medulla means the center. You're gonna see this terminology a lot. So deafness is hearing loss. There's conductive deafness, which is conditions that interfere with the transmission of the vibration. So conductive deafness from the name, we are deaf because there is a failure in conducting the sound. That is curable. You can cure that. You can restore the ability to conduct or better enhance the transfer. You can better enhance the transfer and the conduction of sound by putting on a hearing aid. But sensory neural deafness, that is not a problem of conduction. The conduction is fine. The propagation of the sound wave and its conversion from a sound wave through the ossicles and through the stereocilia is fine, but there is an irreversible damage. There is a damage beyond salvage to the vestibulocochlear nerve, cranial nerve number 8. That one there is no treatment for because you have completely damaged. And we said that neurons don't regenerate. If neurons regenerated, we can make a new cochlear nerve. But because they don't regenerate, that's why we have stroke. If neurons regenerated, we would never have a stroke. Because once the brain tissue died, we can have new brain tissue. But because it's terminally differentiated, quiescent type of tissue, that's why when it's damaged for good. So we're done with hearing. So now we're going to look at equilibrium. Equilibrium is the sense of the body orientation, movement, and balance. And that is very important. We are concerned with our balance, especially when we're accelerating. And you remember from physics, there is linear acceleration. An example of linear acceleration is when you're running or when you're in the elevator. When the elevator goes up and down, that's linear acceleration. So you're supposed to be able to control your balance when you're accelerating forward or backward, or if you're on the elevator, if you have a problem in your balance, patients that have a defect, patients who suffer from an inability to control their balance, they can never get on the elevator because once the elevator starts moving, they cannot control their balance and they will fall, or if they start accelerating. So we can accelerate linearly, or we can have an angular acceleration. What's an example of angular acceleration? Ever spin the chair? Ever been on a chair and you spin the chair? Yeah, that's angular acceleration. No patient with equilibrium deficit can spin on a chair. They will completely lose their balance and they will fall. But you can spin in the chair while still being at it. The chair is spinning, but you don't fall. But if the chair spins with them and they are suffering from an equilibrium problem, from the first spin, they're going to fall off the chair. So your ability to resist falling while the chair is spinning indicates that you are successful in maintaining equilibrium. Your ability to stand tall while the elevator is going up and down and you don't fall, that shows that your equilibrium is intact. Your ability to stand tall while your eyes are closed. Some patients, they have a equilibrium clinical problem if they close their eyes. So they rely on their visual stimulation to acquire their balance. But if they close their eyes, they will immediately fall. That's known as the Romberg disease. We will talk about that. Okay, the saccule and the utricle, they contain a macula. There's the macula sacculi that lies vertically on the wall of the saccule, and the macula utriculi that lies horizontally on the floor of the utricle. So we have static equilibrium and dynamic equilibrium. Static equilibrium is when the head is tilted and the heavy otolithic membrane is going to sag, bending the stereocilia and stimulating the hair cells. Dynamic equilibrium is when the linear acceleration is detected as the otolith is going to lag behind, bending the stereocilia and stimulating the hair cells. Because the macula sacculi is nearly vertical, it responds to vertical acceleration and deceleration. Okay. So how does the physiology of equilibrium how is it achieved? The semicircular ducts, these are rotary movements that are detected by 3 semicircular ducts. The spatial orientation of the canals is going to cause the ducts to be stimulated by rotating in different planes. Each ampulla, so each duct is filled with an endolymph and is going to open up as dilated sac known as the ampulla. And each ampulla is going to contain a crista ampullaris, which is a mound of hair cells and supporting cells. So the projection pathways, how do we project that the chair is spinning? How do we project that we are standing up in an elevator? So all the hair cells are going to synapse on the vestibular nerve. So that's not the cochlear nerve because we're not dealing with hearing. This is the vestibular nerve since we're dealing with the equilibrium. So fibers are going to end in a complex of 4 vestibular nuclei on each side of the pons and the medulla. The left and the right nuclei are going to receive input from both ears, and they're going to process the signals about your position and the movement of the body, and they're going to relay the information to 5 stages. The vestibular nuclei is going to relay the information to 5 areas, and the cerebellum, that is the most important region for coordinated and planned movement. The cerebellum is going to integrate the vestibular information into its control of the head and the eye movement, and the muscle tone and the posture. Reticular formation is thought to adjust the blood circulation and breathing to the postural changes. So that shows us that our ear is also implicated in having first order neurons that regulate our balance. So the first order neuron is going to target the cerebellum to achieve the motor coordination, and that's going to elicit the awareness of the spatial. So now you're aware of the fact that you are in an elevator, you're aware of the fact that you are in a chair that is spinning, and that's going to cause you to resist the consequences of falling. Okay, so we'll do vision on, we'll finish vision on Friday, and we will do the endocrine system on, not Friday, Thursday. So Thursday we will do vision, and we will also do the endocrine system, and exam is Monday. So please make sure to do your homework and go over all of these.